Good morning, my plant-loving friends. Welcome to your Daily Plant Pulse, a podcast-styled series rooted in my deep fascination with plants. Here, plants go through a plant pulse research protocol where we uncover fascinating insights from scientific studies and share them with you. All studies I review are sourced in the description. I'm not a doctor, just someone who loves to research plants and present what I find. Our 9-part series takes you from an introduction to the plant through amazing studies and even an occasional bonus episode. Be sure to check out our Plant Pulse playlist where all of our episodes on all plants will be. And when a plant profile review is complete, explore our Living Library playlist for an in-depth consolidated review. We dig deep into history, forgotten studies, and untold stories so plants can have a voice and their story can be told. Welcome to episode 9, where we dive deep into scientific studies exploring the incredible ways plants can support human and animal health, as well as solve practical real-world problems. There's so much to uncover that this episode will have many extensions. Look for 9.1, 9.2, and beyond as we continue unlocking the hidden potential of plants, largely undiscovered by mainstream society. Until now. So let's dive into one of the most exciting areas of natural compound research, the anti-cancer potential of Diosgenin, a powerful plant compound found in Dioscorea yams, also known as wild yams. Diosgenin is drawing major attention because in both lab and animal studies, it has shown remarkable effects against a wide range of cancers. And what's most interesting is that it does this while sparing healthy cells. And yet, with all that promise, human trials remain scarce. It makes you wonder, what does a compound have to prove before it earns serious attention in mainstream medicine? Today, we'll walk through what the research has found, define the medical terms and molecular players involved, explain how Diosgenin actually works at the cellular level, and even compare its effects to conventional cancer drugs. So by the end, you'll have a clear sense of why this compound is so intriguing and why it's worth asking, why hasn't this gone further? So let's start at the foundation. Diosgenin is what's known as a steroidal saponin. That means it's a molecule made up of a sugar chain attached to a steroid-like chemical structure. This combination is important because it allows Diosgenin to interact directly with the fatty outer membranes of cells with the internal signaling systems that control inflammation, metabolism, and growth. Those are the very systems that tend to go haywire in cancer. And those are also the systems many synthetic drugs try to target at a much higher cost, both financially and physiologically. Scientists have also been experimenting with slightly modified versions of Diosgenin, which they call derivatives. 2 of the most studied are referred to in the research as Compound 8 and Compound 7G. These are essentially refined versions of the original molecule that were designed to make it even more potent against cancer cells, while still being selective, meaning they continue to spare normal healthy cells. It's fascinating work, and again, mostly confined to preclinical labs, not large-scale human studies. Why does funding dry up just when the results start to get interesting? Now, when we talk about anti-cancer activity, we're talking about evidence across many different cancer cell types. Researchers have tested Diosgenin against a long list of human cancer cell lines. That's lab-grown models of cancer that originate from actual human tumors. In breast cancer research, Diosgenin has been tested on several commonly used cell lines. These include MCF7, representing estrogen receptor positive tumors, the more aggressive triple negative MDAMB231, as well as T47D and HS578T. For cervical cancer, studies have focused on HeLa, Cascai, and SIHA cells as derived from human cervical tumors. In liver cancer research, scientists use HepG2, Bel7402, and SMMC7721 cells. Prostate cancer studies rely on the PC3 and DU145 lines, while colon cancers are represented by HCT116 and HT29. Finally, for blood cancers, Diosgenin has been tested on a leukemia cell line called K562. All of these are well-established models used worldwide to test how potential anti-cancer agents behave before they're ever tried in animals or humans. And that's where many plant compounds seem to stall, at the threshold between promising data and real-world validation. Why is it that funding seems to just dry up once it's confirmed that these plant compounds are outperforming many pharmaceuticals? Now, how do scientists measure Diosgenin's potency? They use something called the IC50, for short, inhibitory concentration 50. It's the amount of a compound needed to kill 50% of the cancer cells in a dish. The lower that number, the stronger the effect. For Diosgenin, IC50 values range anywhere from about 0.25 micromoles up to 100 micromoles, depending on the cancer type. To give you context, breast and cervical cancer cells responded to extremely low concentrations, sometimes as low as 1 micromole, which is remarkably potent. Liver cancer cells showed a broader range of responses, while certain Diosgenin derivatives, especially compound 8, were extremely effective against blood cancers, killing cells like K562 around the 4.4 micromole mark. If we rank the cancers by how sensitive they are to Diosgenin, starting with the least affected, it looks like this. Prostate cancers, represented by PC3 and DU145, were the least sensitive, needing the highest concentrations to see an effect. Next, liver cancers, including HepG2, Bel7402, and SMMC7721, showed a moderate response. Colon cancers like HCT116 and HT29 were more responsive. Moving up the scale, blood cancer such as the leukemia line, K562, reacted strongly to Diosgenin and its derivatives. Cervical cancer cells, including HeLa, Cascai, and SIHA, were highly sensitive and at the top of the list. Breast cancer cells, particularly MCF7 and MDAMB231, were the most affected of all. This ranking gives a clear picture of where Diosgenin hits hardest and where future research might focus its attention. And we're all watching to see what happens next, because we're wondering, when are they going to be funded? In terms of how Diosgenin is tested, there are two main approaches. In vitro studies, meaning in glass or in the lab, Diosgenin is simply added to the cancer cells growing in culture so scientists can directly observe what happens. In vivo studies, meaning in living animals, Diosgenin is usually given orally or through an injection in vivo. These animal studies often run for several weeks, with doses typically between 20 and 100 milligrams per kilogram of body weight. While those numbers are specific to animal physiology, they help us understand how Diosgenin behaves once it's absorbed and processed inside a living body. Still, it raises the question, why do so many natural compounds with this level of promise never reach human testing? We're going on 70 years now of this discovery. The first big action is apoptosis, or programmed cell death. The cell's built its self-destruct mechanism. In healthy tissues, apoptosis is what keeps damaged or mutated cells from turning cancerous. But once cancer develops, those pathways are usually turned off, which lets the diseased cells live indefinitely. Diosgenin appears to switch them back on, reactivating the death signal so the cancer cell essentially tells itself to shut down without affecting healthy cells nearby. The second action is called cell cycle arrest. Every cell divides through a sequence of phases known as the cell cycle. Cancer cells rush through that process uncontrollably. Diosgenin puts up a red light halting that division at specific checkpoints, which stops the tumor from growing and gives the body's immune system a chance to catch up and clear out those halted cells. The third major effect is through oxidative stress. Diosgenin increases the levels of reactive oxygen species, often called ROS, inside cancer cells. ROS are unstable oxygen molecules that can damage DNA, fats, and proteins. Cancer cells are already under high oxidative stress because they grow so fast and burn energy inefficiently. When Diosgenin turns up that pressure, the cancer cells can't handle it and they die off. Meanwhile, healthy cells stay safe because their antioxidant defenses are stronger and can neutralize the extra ROS. Diosgenin also targets several key molecular signaling systems that cancer cells depend on. It lowers levels of matrix metalloproteinases, often abbreviated as MMPs. These are enzymes that help cancer cells break down surrounding tissue so they can invade and spread. It blocks the AKT pathway, a survival signal that keeps cancer cells growing even when they shouldn't. And it suppresses NF-kappa B, short for nuclear factor kappa light chain enhancer of activated B cells, a master switch that turns on genes linked to inflammation and cell proliferation. By shutting down NF-kappa B, Diosgenin helps calm inflammation and stops the runaway growth signals that cancer depends on. That's especially exciting. Diosgenin's selectivity across many studies, it shows what scientists call selective cytotoxicity. That means it kills cancer cells but spares normal ones. Its derivatives compounds 7G and 8 are even more potent but still display the same safety profile. In animal studies, no toxic dose has been identified so far. Isn't this the cancer cure that pharmaceutical companies have been searching for? So again, we have to ask, it's been 75 years since we have known of its anti-cancer effects. How many billions, trillions of dollars has been spent on anti-cancer research, curing cancer? How many billions and trillions of dollars have been donated to these causes? If it's that safe, that targeted, and that effective, why isn't it already being tested in people? And why isn't society demanding more? That's in a sharp contrast to most chemotherapy drugs, which attack all fast-dividing cells, healthy or not. Why is it that chemo often causes immune suppression, nausea, and hair loss? Because it's being given when alternative options are out there waiting to be researched. Chemotherapy hits bone marrow, digestive tissue, and hair follicles along with the tumor. Diosgenin, on the other hand, seems to recognize the difference, focusing its attack where it's needed. So what does all of this mean in practical terms? Diosgenin acts on multiple fronts. It stops cell division, damages the DNA of cancer cells, ramps up oxidative stress, where the tumor is vulnerable and switches on self-destruct mechanisms that cancers usually silence. And it shuts down the genes that drive invasion and inflammation. The doses required are tiny, and the animal data confirm that it works in a living system, not just a petri dish. Now, it's important to remember that this is still preclinical research for some reason. That means these findings come from a lab in animal studies, not yet from human trials. We don't know yet what a safe or effective dose would look like for some reason. But the groundwork is promising, and the consistency across many different cancers suggests Diosgenin may eventually play a role as a supportive or complementary therapy once clinical trials are done. If, of course, those trials are ever funded. That's the lingering question. With so many natural compounds, what determines which ones get their turn? So to recap, Diosgenin, this natural, wonderful compound from Dioscorea yams that show potential selective anti-cancer activity across breast, cervical, blood, colon, liver, and prostate cancers. It works through apoptosis, cell cycle arrest, oxidative stress, DNA damage, and inhibition of survival pathways like MMP, AKT, and NF-kappa B. It's selective, non-toxic in animal models and stands out from conventional chemotherapy because it targets cancer's weak points while protecting healthy cells. When you step back and look at Diosgenin's bridges, it bridges ancient plant wisdom with modern molecular science. It represents how compounds from simple roots quite literally can teach us new ways to fight disease. But maybe it also represents something else, a blind spot in how modern medicine decides what deserves attention and what gets left behind.
And that brings us to the end of our look at the studies behind this remarkable plant. We've explored the science. Now it's time to see where the path leads next. Sometimes this marks the close of a chapter, and sometimes it's just the beginning of another. Thank you for following along and being part of this growing community. If today's research sparked ideas or questions, I'd love to hear them in the comments. And if you're as excited for what's next as I am, make sure to like, subscribe, and tap the bell so you don't miss the upcoming episode. Together, we're turning information into understanding, and until next time, let every moment bear fruit. The pulse continues.